All right, so we're going to start off with reviewing the white blood cells. We'll get into the lab values and and then review again the blood typing. Even though we did that in lab, I just want to go over specifically the role of the D antigen, since I didn't cover that in the lab component. So with white blood cells, so we've already talked about red blood cells. I'm going to tell you to review in your textbook, to review the things that we've already covered. I know I've transitioned to these new slides, so hopefully you will review those slides, and I'll let you know if there's anything specifically to review. And these are the slide sets that we'll use from here on out. Now, this slide set does have some additional information. You won't necessarily need it. I just wanted to give you a visual instead of just talking about these things. These are things that you'll be able to get from your lecture book. So, all right. So, leukocytes and white blood cells. Your white blood cells are one of the things that they're primarily known for is their function in defense against diseases. So, think of viruses, bacteria, fungal infections, things along those nature. But they also play a role in inflammation, which may or may not have a role with in any kind of infection. The other thing that white blood cells are known for is they actually help, particularly the macrophages, they kind of help with just the breakdown and the wear and tear of the different elements of the body. And they help to keep and get rid of old cells that are no longer needed. So they play a role in not only defense against diseases, inflammation, but also in just kind of like the regular maintenance of the body. So they are very important. They're the cells that are constantly monitoring the health and stability and function of the different tissues. They're the ones that kind of are going to help the body to identify when there's a problem state or disease state, because it'll actually start to pick up some of the remnants of the breakdown of tissues, and then they go into the lymphatic system where they process that. So we'll talk more about the lymphatic system when we get to that chapter. So a couple tidbits about the white blood cells. They're able to leave the bloodstream and actually go into the tissues, and we call that process emigration. You could also refer to that as migration, but basically what they have the ability to do is they can actually leave the bloodstream. Now, your red blood cells don't leave the bloodstream. At least they're not supposed to. The only reason they would leave the bloodstream is if there was a rupture in the blood vessel. You will discover that a little bit more clearly when you review the Anatomy TV section on hemostasis. Again, they act as a signal for inflammation and tissue repair, and they use the cell chemical structures known as cytokines. So there are two main categories. You have granulocytes and agranulocytes. The granulocytes mean that there are visible cytoplasmic granules. So if you look here on the left of the slide, these are your granular leukocytes, and it's because you can actually see their cytoplasmic granules quite easily. Your agranulocytes says there's no visible cytoplasmic granules, but if you look here, you actually do see the cytoplasmic granules. They're just much smaller in size than the granulocytes. So both granulocytes and agranulocytes do have cytoplasmic granules. It's just that the granulocytes are much bigger. So your granulocytes are your benphils. Those are your basophils, your eosinophils, and your neutrophils. Your agranulocytes consist of the lymphocytes. So you have B lymphocytes and T lymphocytes. And then there's the monocytes. Okay. And then you can see this last note here. Less than 1% are going to be found in circulation. Most are going to be found within the lymphatic structures or in the tissues around the body, such as surrounding the liver, the lung, the skin. You actually have specific areas known as your lymphatic tissue. So we'll talk about the mucosal associated lymphatic tissue, the gut associated lymphatic tissue, and that's where you're primarily going to find your white blood cells. So let's look at the different white blood cells and their functions. So the first one here is the neutrophil. The neutrophil is going to be indicated and will increase its numbers during most, if not all, types of infections as well as inflammation. Their role is to phagocytize, so to like break apart and then release hydrolytic enzymes. So remember, phagocytosis is the process of the cell eating or breaking down another, let's just say, another cell. And then the hydrolytic enzymes are enzymes that are going to also further break it down. So it's kind of like these neutrophils have like acidy spit that they're shooting out at, let's say, bacteria in order to help break down that bacteria. So neutrophils tend to be identified as what are known as the bacterial slayers, meaning that when bacteria is present, neutrophils tend to come up and they increase in the number that are present in order to take on the bacteria. They are also the primary components of pus. So a lot of times in pus, you'll find dead matter, you'll find a lot of liquid, and then there's also going to be dead neutrophils in that area too, because they basically went in there to kill the bacterial slayers, and then it pushes it out through your skin so that it can eventually be released and get out of the body. So that's your neutrophils. Now, notice and we mentioned this before in a previous lecture, your neutrophils are considered polymorphonuclear, meaning that they have multiple lobes or lobules for their nucleus. So it's called polymorphonuclear. Eosinophils, remember they're the reddish or pink staining granulocyte. They tend to increase in their number when there are parasitic infections as well as allergic reactions or allergies. So a parasitic infections could mean like worms. So if you remember the movie Dumb and Dumber, and he says proudly that we've got worms or I've got worms, indicating his worm store that he wanted to open up. Well, if he had worms and he had intestinal parasitic worms, he would also see an increase in the eosinophils present. Also, the role of the eosinophils in allergies is actually their job is to reduce the allergic reaction. So we refer to that as mediation of the allergic response. So eosinophils being released helps to modulate or reduce the inflammatory response associated with allergies. So they kind of counteract the ones that are also involved with the allergic response, which is your basophils. Now remember, your basophils are like the darker purple stain or darker blue staining granulocyte, and they're going to be very, very important in mediating the allergic response, sometimes to the detriment of those of you who suffer from allergies. A lot of times you could blame your basophils, and then a cousin of the basophil is the mast cell, M-A-S-T, mast. So basophils secrete histamine. Histamine is a potent vasodilator, meaning it opens up the blood vessels. It increases blood to that area, which causes redness and swelling, which is typically associated with allergic reaction. Like if you think of like a bee sting, or even if you just get like a mosquito bite or something along those lines, you basically are seeing histamine release in action because the site gets warm, the site gets itchy, the site gets swollen, and all those are characteristics of histamine. Basophils also secrete heparin, and heparin is an anticoagulant, and you'll review what anticoagulant is when you go through the Anatomy TV section on hemostasis, but basically it prevents clotting, and what that does is it makes it easier for red blood cells and other white blood cells to enter into the area. And again, sometimes this system is a little bit out of whack, meaning out of balance, and that helps to explain why some people have extreme reactions or allergies, like seasonal allergies, hay fever, or even to the point where they could have anaphylactic shock as a result of a bee sting or consumption of peanut butter or other things, or shellfish. A lot of that has to do with the stimulation of the basophils and the mast cells, the histamine response, and the role of heparin. And so just trying to tie that into the allergic response. And it's also why not everyone has the same types of allergies. It's because it varies from person to person. Your A granulocytes, you have lymphocytes and monocytes. This is your lymphocyte here. Has a very large nucleus, takes up, and you can just see like a little thin layer of cytoplasm surrounding it. It consists of the T cells and the B cells, meaning that they're involved with your specific cell-mediated / antibody-mediated type immunity. And so T cells tend to be more like they go in and they interact with the cells. Let's say the cells that are virally infected cells or bacterial cells. And then B cells are more involved with secreting plasma cells, and plasma cells secrete antibodies. So remember, we talked about antibodies are the gamma globulins, the immunoglobulins that are secreted, and they contribute to the proteins in plasma, but also they play a role in what's known as antibody-mediated immunity. The T cells and the B cells are, if you've been obviously following along with the what's been going on with the COVID pandemic, it's the T cell and B cell memory cells of both that we're trying to identify, or scientists are trying to identify which and if there can be any kind of immunity after having either natural or artificial exposure to COVID-19 or its components. So that's what they're still investigating and learning and hopeful for. A lot of times people think that it's the antibodies, but sometimes you do also have T cells that have memory, and that's why you may not have antibody mediated, or you may not have antibodies for COVID, but you may have protection because of the T cells. And then I think the lab values for that are just harder to test. So this probably explains why some people test positive and have no symptoms. Some people test positive and have very mild symptoms. And then some people test positive and have very extreme symptoms. A lot of it might be the roles of the different T cells. More so than, I know somebody brought up the different blood types. It may have more to do with T cell function and B cell function than that of just red blood cell function. So again, I don't know that specifically. I'm just making a hypothesis about some of the aspects that I've read about these things, and then just trying to tie it into the lymphocytes, or to COVID. So when there's an increase in the number of lymphocytes, it typically can mean any number of things. It's, usually it's, you're going to see elevated lymphocytes with viral infections, but other things could drive those to go up. Lymphocytes are also play an important role in destroying certain cells that have been identified as being cancerous. Foreign cells, so like if you give somebody the wrong blood type, it's your lymphocytes that play a role. And then the antibodies that play a role in attacking those foreign cells. And then, as I mentioned earlier, virally infected cells is where you typically see lymphocytes elevated. Monocytes tend to increase in numbers during viral infections as well as in inflammation. They also are known for leaving the bloodstream, and once they get into the tissues, they're known as macrophages. So you may remember in the liver, I mentioned specifically Kupffer cells, Kupffer, Kupffer cells, that are going to be found within the liver. But at one point, those macrophages or Kupffer cells in the liver were circulating monocytes. And their role, their macrophages tells you macro is big, phage is eater. So these are your big eaters, and they are going to eat up pathogens and debris and break them down. So the monocyte is very large. It has like a kidney-shaped or kidney bean-shaped nucleus, and then it's just a way to differentiate what that looks like. So some disorders of the leukocytes can include leukopenia. Leukopenia just means a low or abnormally low white blood cell count, and this can be due to a number of things. It could be due to radiation therapy. So when someone's receiving radiation, one of the things it does is it decreases the white blood cell production. That's actually why they use chemotherapy and radiation in cancer patients, is they're trying to decrease mitosis or mitotic divisions. And so if someone gets radiation, let's say from not a therapeutic reason, that can cause their white blood cell counts to drop. This is also why someone who's receiving radiation or chemotherapy also loses their hair, is because hair is constantly going through mitotic division, and basically when you are stopping that process, you're stopping that process everywhere. So when you understand that, it actually helps you to understand what someone is going through when they're doing that. It's also why people who end up going through like chemotherapy or radiation therapy may want to store their gametes, whether that's sperm or egg, because there's a possibility that they become sterile after going through chemotherapy or certain types of radiation. Certain poisons can drop leukopenia, certain types of infectious disease, and I have a slide coming up that will just kind of highlight some of those. When someone has leukopenia, and it's because of some of these other reasons, it actually increases the risk of different types of infection. So think about it. If you have less numbers of white blood cells than you should have, it's going to leave you open to possible infection. So a good example of that would be, it's kind of like you have this fence around your house that's there to protect you from, foreign, or not foreign, but protect you from invaders. But what if you have the gate open? Or what if parts of the fence are missing? It just makes it easier for those things to get in and potentially wreak havoc. What about the opposite, leukocytosis, where you have an unusual or abnormally elevated white blood cell count? That could be due to certain types of infection, certain types of allergies, and certain disease states can certainly drive up the white blood cell count. You would think that maybe you would have more protection, but a lot of times it really just indicates that there's an active inflammatory or infectious process going on. Sometimes that's acute, meaning short term, or sometimes it's chronic, meaning long term. And that's where you'll see this in a little bit later, is a differential white blood cell count comes into play. It helps to identify the percentage of the total white blood cells, and then helps us to differentiate what the problem might be. So let's just say we know a given percentage for neutrophils, and let's say those neutrophils are elevated. Well, what were neutrophils known for? They were known for bacteria, right? For attacking bacteria. So you might assume that if the neutrophils are elevated, that there might be a bacterial infection. You could also assume that it's inflammation as well, but when neutrophils are elevated. Or what about if eosinophils are elevated? What are you thinking? You might be thinking parasitic worms, right? Now, there's actually a way to remember, there's a mnemonic to remember the leukocytes in order of what their in their level of abundance, and the term is, or the thing to remember is, never let monkeys eat bananas. Never let monkeys eat bananas. So never refers to the neutrophils. That's the most abundant type of white blood cell. Let is the lymphocytes. That's the second most abundant type of white blood cell. Monkeys. So monkeys is monocytes. That's the third most abundant type of white blood cell. Eat, eosinophils. That's the fourth most abundant type of white blood cell. And then bananas is basophils. That is the least amount of, or the fifth most abundant, or the least abundant type of white blood cell. So never let monkeys eat bananas is a way to put those in order. And to remember which ones are typically higher and lower. Now, I'm not going to ask you specifically, at least not on here, or to know on like a test. It may ask the question on a quiz, but you know your quizzes are open book. I'm not going to ask you to memorize percentages, but it is helpful to know what is the most abundant and then down to the least abundant. And so, never let monkeys eat bananas will help you with that. Mononucleosis, you may have known someone who's had mono, or maybe you've had it yourself. It's a viral infection that actually elevates the number of lymphocytes in the blood. So you will actually see a specific number of lymphocytes. What is mono? Well, mono is a viral infection. So of course we're going to see lymphocytes increased in a viral infection. What about leukemia? So we talked a little bit about leukemia. I talked about how the way that they differentiate the different types of leukemia is depending on the cell lineage. So if it's myelogenous, that means it's typically all of the white blood cells, excluding lymphocytes, and then the lymphogenous would be all the lymphocytes. And so that's how you differentiate the two main types of leukemia, and then it's either acute or chronic. But basically, it's a cancer that's affecting the development of the white blood cells in the bone marrow. Because the white blood cells being produced are abnormal, they don't fight off infection, so it leads you to being susceptible to infection. And then also, because of the excess number of white blood cells, it can actually get into, invade, and destroy some of the normal tissues that are there. And one of the things I gave you was, it doesn't necessarily destroy these areas, but because there's so many white blood cells being produced that are abnormal and the body's trying to remove them, it actually puts an extra stress on the liver and the spleen. If you remember, I used the term hepatosplenomegaly to identify where somebody would have pain in their upper quadrants of their abdominal cavity, the right upper quadrant and the left upper quadrant, because that's where the spleen and liver are located. And that swelling means that the spleen and the liver don't function normally, and it's usually an indication that they are processing too many cells at any given time. The other thing that you will see associated with leukemia is because there's such an abundance of white blood cells being produced, what that does, it actually shuts down or downregulates the production of other types of cells, including red blood cells and platelets. So sometimes an early sign or symptom of leukemia might be abdominal discomfort, and it might be they might get like a cut or a scratch, and then it doesn't clot or doesn't stop bleeding. It just continues to bleed. That could be an indication of leukemia. All right, so we have when they do a blood test, they'll do a CBC, which is a complete blood count. So they're pulling up everything. They will look at different values, including hematocrit, hemoglobin concentration. They'll look at the red blood cell counts. They'll look at reticulocytes, the white blood cells, the platelets. They'll do a differential white blood cell count. And then they're also going to look at the size of the red blood cell as well as the concentration of the red blood cell. So let's look at these a little bit more specifically. So your red blood cells, you can do an RBC count or red blood cell count, and that's just going to measure the total number of red blood cells in a microliter of blood. Remember, if the red blood cell count is too low, we might assume that someone is anemic. If it's too high, we might assume that someone has polycythemia, and we discussed those already with the red blood cells. We're also going to look at the percentage of reticulocytes. Remember, I told you that range can go from 1% to 3%. So the normal range is around 1%, but if it's increasing, meaning that we see increased number of reticulocytes, that typically might indicate an increased rate of red blood cell production. So that could be an indication that someone's at a high altitude, or it could be an indication that somebody is blood doping, so they're using EPO. Hemoglobin is the measure of hemoglobin content of the blood, so that's just another way. So you could have a normal number of red blood cells, but if you don't have enough hemoglobin, then you have diminished oxygen carrying capacity, and that's also a characteristic of certain types of anemia. And then finally, hematocrit, that's actually the mass of the red blood cell. So how big is the red blood cell? So let's look at these a little bit more specifically. So when we talk about hematocrit, there's another term associated with that, and it's called the mean corpuscular volume, or the MCV. And this is actually specifically the volume or the size of the red blood cell, and it can be identified a couple different ways. If it's normal, we would say it's normocytic. That's the size of the red blood cell. It's normal size. If it's too small, we would say it's microcytic. And if it's too big, we would say it's macrocytic. So it's just a way to kind of differentiate the size of the red blood cell. We also have a way to differentiate the color, and what determines the color, remember in your hemoglobin, you had those 4 chains, the 2 alpha and 2 beta chains, and that's what contributes to the color of the red blood cell. Keeping in mind that when there's oxygen on the hemoglobin, you are going to see a bright red color, and then when there's no oxygen, you're going to see more of a flat red or a dark red color that's not as bright. Okay? So the mean corpuscular hemoglobin concentration is what that term is referring to, and we look at the color. So if the normal color, we would just say it's normochromic, has a normal color looking to it. The decreased color, meaning that if it's lacking color, it's less red, we would refer to it as being hypochromic. Okay, and then this is just a mistake. I left this slide here, and so just ignore this. This is actually from the previous slide. I just forgot to delete it, so sorry about that. So those are your red blood cell considerations. So we can use what we know now to identify different things. So this is my normal red blood cell. We would say this is normocytic, okay, and normochromic. So this is what we expect to see. Now let's go up here. Look how big this red blood cell is compared to these down here. So we would actually say that this is macrocytic, okay, but the coloring looks the same, so we would say it's normochromic. Now, macrocytic type anemias are typically identified as megaloblastic anemias, and they're usually due to B vitamin deficiencies. Okay. What about an iron deficiency? Let's say someone's lacking iron. So if they're lacking iron, that means they can't get the oxygen. So there's two things that are going to occur. First of all, if there's no iron there, then the cells itself are going to actually be smaller because there's less content there. So this is a microcytic. And then you can see how the color doesn't look nearly as bright red as the normal red blood cells. So we would say this is hypochromic. So iron deficiency type anemias tend to be microcytic and hypochromic. And then finally, sickle cell disease. Notice the color is normal. Okay, so we'd say normochromic. You could say microcytic, but then you could also just identify the fact that it's sickled appearance, and so it has a very different appearance than what it should normally look like. So it has that like sickled, spiral shaped, that basically tells you that there's a problem with the hemoglobin synthesis. It also tells you that it doesn't carry oxygen very well. So those are your red blood cell considerations, or your red blood cell lab values. Let's look at the white blood cells again. So we already mentioned these: leukopenia, low white blood cell count, but I told you I would tell you the specific things involved with that. So that could be associated with typhoid fever, the flu, measles, mumps, chickenpox, and then of course AIDS. All those can drive or lower white blood cell counts. High white blood cell counts, we talked about leukocytosis, we talked about mononucleosis associated with that. It's also associated with acute infections, vigorous exercise. So like after a really intense exercise regimen, you might actually see elevated white blood cell counts, or significant loss of body fluids. So if somebody has they're losing blood, you might also see an increase in white blood cell development as a result of the process associated with replacing or responding to that loss of body fluid. And then we already talked about the differential. It's going to be based off the percentage of the types of leukocytes. It will change with different pathologies. So if, like I said, if somebody has a parasite infection, we might see an increase in the number of eosinophils. If they have a viral infection, we might see an increase in the lymphocytes. If we have a bacterial infection, we might see an increase in the neutrophils. So it's going to be dependent on what the infection is, if it's inflammation, what's going on with the body. Another white blood cell concern is neutropenia, where there's actually an abnormally low number of neutrophils, and this can be due to a number of things. It could be because there's a decreased actual production, or there's defective production. So that can happen because of certain types of drugs, certain types of medications can actually lower neutrophil production. Neoplasms, which is cancer, can also cause a decrease or defective production of neutrophils. Specifically, leukemia would be a good example of that. There could also be congenital considerations for a decrease in the number of neutrophils, meaning that maybe genetically these people just naturally produce a low number of neutrophils. So that doesn't necessarily mean it's bad. It would be bad if they're also having and increased risk of infections. Sometimes the body does some amazing things to kind of compensate when things are lower or higher than expected. There could be impairments associated with the production. There could also be an increased rate of destruction. So you might actually see that associated with certain types of inflammation, certain infectious infections, and then autoimmune disease is also associated with driving down or decreasing the number of neutrophils that might be present. All right, so we already talked about blood typing in lab, and so I'm not going to go through all these again, other than just to remind you that, remember, the antibodies attack antigens. So when your plasma cells secrete antibodies, they're designed to go after whatever antigen they are unique for. So let's say I have E. coli, and E. coli has the E. coli antigen. I'm going to have anti-E. coli antibodies released by plasma cells that are going to attack certain types of E. coli. Okay? So in blood typing, the A antigen is found, and it's normal, and it's supposed to be there. All of our cells have antigens. They're just the things that indicate, hey, I'm Frank, I'm Dave, I'm A blood, I'm B blood, I'm AB blood. It's the presence or absence of the antigens that gives it the cells their specific names and types. Later on, when we talk about the immune system, we'll talk about CD4 cells, CD8 cells, CD28 cells, and those are just basically different antigens that are found on those white blood cells that indicate, is it a T helper cell, is it a T cytotoxic cell, is it a memory T cell, is it a memory B cell, and we'll be able to differentiate those different cell types based off of their antigen, okay? So the antibodies are going to attack things that shouldn't be there. So when someone has type A blood, they're going to secrete anti-B antibodies because they don't want blood from B blood types or AB blood types. If there's a B antigen present, the anti-B antibodies are going to attack it. Same thing goes for the B antigens. If there's a B antigen present, and they're going to secrete anti-A antibodies, and they're going to attack any blood that has A in it. So that includes A types and AB types. Okay. Now, the fact that O's don't have antigens, that's why O is the universal donor. Okay. Because AB blood types do not have antibodies, that's why they are the universal recipient. Okay. Now, because O doesn't have antigens, that's also why they have both anti-A and anti-B antibodies. It's those antibodies that are specific for attacking other blood types, such as the A blood types, the B blood types, and the AB blood types. So if you gave type O blood person type A blood, they are going to reject it, okay? And it's because of their antibodies, okay? Now, the other thing that's important is, as these red blood cells develop, these antibodies also are released and developed. So you don't have to have previous exposure to B blood type or AB blood type B or AB blood type in order to secrete anti-B antibodies. What I mean by that is, a lot of times when you get, let's say you get a certain type of infection, your body, in the initial infection stage, will create an immune response, and it'll also create antibodies specific for that infection. It's one of the reasons why, if you had colds more frequently when you were younger, but then as you got older, you don't have the same colds because your immune system already knows how to deal with them. They already fight them off, and then you've already created antibodies to deal with those types of infections. Now, your red blood cells are unique in that they are going to start to secrete the anti-B antibodies at the same time that that the red blood cells are being produced. So there's no previous exposure that's required for these anti-B antibodies. The reason I'm mentioning this is because in a moment, when I talk about the Rh factor, the Rh factor is important and there is exposure involved in order to create what's called the anti-Rh antibodies. So we're going to skip down to those. You can review these because we already did this in lab. We talked about the universal donor and universal recipient. All right, so the Rh group, contains different types of agglutinogens that were originally discovered in the rhesus monkey. Okay, so you can see that there's RHC, D, and E, so there's different RH factors, but the main one and the most reactive is considered the RHD, and that's what gives us is someone positive or negative. If they are RH positive, that means they have the D antigen on their red blood cell. So like for me, my blood type is A positive. So I have the A antigen. I also have the D antigen. Okay. If I were A negative, I would have the A antigen and I would not have the D antigen. And that's how you differentiate negative from positive. Is the D antigen present? Yes, positive. Is the D antigen absent? Yes, negative. Okay. So, RH frequencies vary among ethnic groups. For in the United States, I believe the numbers approximately 85% of people are considered RH positive, and then that leaves 15% being considered RH negative. When someone is RH negative, and if they are exposed to someone with RH positive blood, they will start to secrete what is known as anti-D or anti-RH antibodies. Okay. Now, again, you can see these are not normally present. They only become present once someone who has RH negative blood or their negative blood type is exposed to RH positive blood. So when might that occur? Well, that typically can occur when a mom who's pregnant is RH negative and gets exposure from her child that's RH positive, or by receiving blood transfusion that contains RH positive blood. Now, typically, what happens is, like I said, they're not present initially, but it's almost like that E. coli infection that I mentioned. You don't automatically create antibodies for things, that you've not been exposed to. It's not until you get exposed to them that you actually start to create the antibodies. The exclusion to that is the A antibodies, the B antibodies that are associated with the blood types. That's just a different mechanism. So the anti-D antibodies don't normally get created until someone that's RH negative is exposed to RH positive blood. Typically, because this happens in associated with a fetus, there's typically no problem associated with the first transfusion or pregnancy, but now that they've been sensitized, now they're going to have a problem if they receive that. So let's say someone with RH negative blood receives a second RH positive blood transfusion, they're going to reject it and they're going to have complications as a result of it. The first baby will be born, but future children may not survive the pregnancy because of the RH factor considerations. So what that is referred to as, it's actually called hemolytic disease of the newborn. And this is what happens when mom is RH negative and has a baby. And then after she's had a baby, she's now been sensitized. And so on subsequent babies, mom's blood will actually attack baby's blood in utero. And this was a common problem prior to the 1940s, where, a woman would have a child and then try and have more children and just would have stillborn after stillborn after stillborn, or would have trouble with just holding on to a pregnancy. And a lot of that had to do with the fact that literally mom's blood was attacking and destroying baby's blood. Okay. So what they do now, and what they've been doing since I think the 1950s, is they've created something called Rhogam. Rhogam is given to, I believe it's given 3 times during a woman's pregnancy. And what it does is it prevents the production of RH negative antibodies. It also will bind to the fetal agglutinogens in her blood. So again, she will not form the anti-D antibodies. And what that does is it prevents, and then she can have children without issue. So this is only a problem for moms who are RH negative and obviously babies who are Rh positive, but you won't know that the baby is Rh positive unless you do a blood type or after they're born. If dad's Rh negative, that doesn't matter. If the baby is Rh negative and mom is Rh positive, again, not a problem. It's only a problem when mom is Rh negative. Okay? So that actually